Well, thank you very much for that very nice introduction, both of them. It's a pleasure to be here on such a beautiful day. I'm sorry that you're all here, not experiencing it, but I'm grateful that you came. It's always a pleasure for me to come to Copenhagen because we have a connection through Birmingham in England, but actually I worked here for half a year. I'll refer to that briefly. In the 1980s, had an extremely productive time here and worked with Professor Richard Agel, who may be, I think, in the audience. I haven't seen him yet. And, he's waved. So it was one of the nicest periods of my life. So what I want to talk about today is work in my laboratory that led to understanding how the reproduction of cells is controlled. And I want to begin by giving you an introduction to that problem to explain why it's important both for biology and for understanding how life works and also for medicine. I want to explain how a problem like this, quite a complex biological problem, can be investigated, even when very little is known about it before such studies, and that will put an emphasis on the power of genetics. But I hope you'll also get some sense of how science is done, the importance of teamwork and collaboration, and maybe also how science is an exciting adventure, and I want to try and convey some of that excitement too. So I want to begin by indicating to you the importance of this problem. And it starts really with the cell. And you've already heard about cells. And here you will see the microscope used by Robert Hooke, one of the founders of the Royal Society. And he took a razor and he cut a piece of plant material. It's a piece from a cork, and looked at it under a microscope. And this is a drawing that he published in 1665. And you'll see here a nest of boxes. And this is the first illustration of cells. This is published 350 years ago and is in the first observation that the cell is the structural basis of life, because over the subsequent century or so, cells were discovered in every living thing. But cells are not only the basic structural unit of all life, important as that is, but also, as was shown in the mid-19th century, that it's the basic physiological unit of life as well. And this came from work from a number of investigators, but well stated by Rudolf Virchow, the founder of pathology, who he was a German scientist, in 1858, and this is an English translation, of course, said, "Every animal appears as a sum of vital units, each of which bears in itself the complete characteristics of life." What does that mean? What it means is that the cell is not only the basic structural unit of life, but it is also the physiological unit of life. It is the simplest entity that demonstrates life's properties. He also stated clearly that cells arise from pre-existing cells. In Latin, omnis cellula e cellula, meaning all cells from cells. This, of course, was in an era when spontaneous generation was still being talked about. And this occurs by the process of cell reproduction, which I'm going to talk to you about during my talk. And this here you'll see is a picture of a dividing cell. It's a dividing cell derived from your bodies. It's a human fibroblast cell. And you can see here this dividing into two masses. And this is the basis of all reproduction of all living things. Now, why is it that this process is important? Well, there's three reasons I want to give to you. The first I've already hinted at. It's the basis of all growth and reproduction. I'll say a little more about that in a moment. The second, somewhat less obvious, but actually already indicated in a very nice introduction, it links the molecular replication of the hereditary material, that is the DNA double helix of Watson and Crick. It links that copying to biology, to the division of a cell, because the molecular replication of DNA is of no consequence unless it's linked to biology. The biology link is through the cell division process. So that's the second point I want to make. And the third is, as has already been indicated also, is that it is crucial for cancer. So let's just look briefly at some of those three things. The basis of all growth and reproduction. This is a mouse embryo here, and if you can see clearly, there's little black spots here, each of which is an individual cell. And that was derived from a single cell, a fertilized egg, undergoing repeated cell reproduction, what we call repeated cell cycles, to produce, first of all, a blastula, a mass of cells, an early embryo, and then subsequently the early mouse embryo you see here. It is cell reproduction, it is progression through the cell cycle that is the basis of all growth of all living things and also the basis of reproduction. So that's the first reason why the cell cycle is important. Incidentally, if you aren't convinced by that argument that cells are interesting, I will just remind every one of you in this room that you once all looked like this. This is an egg about to be fertilized, and all of us were once a single cell. And if I haven't managed to capture your interest yet, it's time to leave. The second reason is this linkage between DNA replication, what we call S phase in the cell cycle, and the segregation of those replicated molecules to a process of mitosis. This is the cell cycle, and we're going to talk about this a lot today, so I'd like you just to learn some of the words that we'll be talking about. Every time a cell undergoes its cell cycle, and that is the series of processes that occur from the birth of a cell to its subsequent division into two, this series of events leads to reproduction of the cells, and there are two key events during the cell cycle common to all cell cycles that are essential to all cell cycles. And they are S phase, when the DNA is copied, the Watson and Crick unravels and is copied, and that occurs earlier on in the cell cycle. And once we have two chromosomes in this simplified organism, the second phase, which I mentioned links it to biology, is the segregation of those replicated DNA molecules, which make up the chromosomes, into the two newly divided cells. If these processes do not occur correctly, then you will not inherit a full complement of genes in the two newly divided cells, and the cells will not survive. So these two events have to occur precisely, they have to occur in this sequence, and they have to be perfectly controlled. And there's two other symbols up here I want you to note. One of them is called G1, the other G2. G1 is the gap at the beginning of the cell cycle, after cell division before S phase. G2 is the second gap after S phase and before mitosis. So that's the second reason why this process is important. The third one, by the way, I hand draw these things. If you're looking for beautiful PowerPoint stuff, you're not going to get it, okay? This is what we have to deal with. My graduate students laugh at me, but it's all I can do. And this is to illustrate that if that cell reproductive process goes wrong and you give rise to uncontrolled cell reproduction, this is will lead to cancer, one of the major diseases of the animal world and of humans in particular. What we have here is a sheet of cells, normal cells in blue, and then a damage occurs to cancer-forming genes called oncogenes, and this red cell goes out of control, and we get a mass of red cells disrupting the tissue of normal blue cells, and this is the basis of a tumor. Uncontrolled replication and reproduction of damaged cells that will lead to cancer, and the outcome of that is uncontrolled cell reproduction. So the control of this process is really quite key. So that's the reason why cells are important. That's the reason why cell reproduction and the control of cell reproduction through the cell cycle are important. And these are the processes that I want to talk about today. How is cell reproduction controlled? How is it you can investigate a process such as this? And this work that I'm going to talk about has actually gone on since the 1970s, so some of it's quite old, and much of it was done in this institute here, where I'm still working. It's in central London, and it's a cancer research institute, and I was working mostly at the time in these 3 bays here. And you can see it was quite a long time ago. Just look at those cars. And so much of what I did was here. It wasn't the only place. I also worked in Bern, Switzerland, in Edinburgh, in the University of Sussex, briefly here in Copenhagen, and in London and Oxford. This is because I couldn't keep a job down, so I had to keep moving. But in the end, it all worked out fine. So let's now imagine turning the clock back to around 1970, it's a long time ago, 40 years ago, and let's imagine you have this problem. The problem is, how do we investigate a complex process such as how the cell cycle is regulated? Well, the approach that I want to describe is a genetic approach. And the beauty of a genetic approach is that it allows you to investigate a problem about which you know nothing when you start. And that is a crucial step forward. Eventually we will get into molecules and how they work, but if you don't get to the right molecules, it doesn't matter how beautiful the molecular biology is, it is of no relevance. You have to know what your molecules are doing, you have to know where to look. And the approach that geneticists use are to take an organism which is genetically amenable, one that you can study, and the one that I use is an organism called the fission yeast, which Richard Agel was an expert on when I was learning how to work on this, an organism called the fission yeast, which is simple. It only has 5,000 genes, we have 25,000. All it does is grows and reproduces itself. It doesn't have to make eyes and noses and ears and legs and so on. So it's stripped down to the problem that you're interested in. Having chosen a simple genetically amenable system, what you can then do is look for mutants in genes that may be relevant to the process that you're interested in. So you can take yeast cells, you can treat them with a chemical, for example, that will damage DNA, and then look for yeast clones that are derived from that, which show a defect which is informative about the process that interests you. And that I've tried to schematize up here. This is a yeast cell. These are a number of genes, these sort of green blobs here. Hit it with a mutagen. Before the mutagen damages it, the cells grow and divide perfectly normally. Now, what you have to do is try to imagine what a cell would look like if a gene important for the cell reproductive process becomes damaged. So the first step in this is a step of imagination, And that step is to say, right, if a cell cannot reproduce and cannot divide itself, but it can continue to grow, that is to make protein, RNA, make mass, then, very simply, instead of looking like this, which is a short cell which is undergoing division, the cells will continue to grow, but they don't divide. And so what you could look for are mutants which generate very long cells that can't divide. It's very simple, but it is a feat of the imagination just to imagine what it looks like. Of course, if they never divide, they will always be dead. So you have to look for a condition where they are normal and where they're defective. And one of the conditions, again also already mentioned, is temperature. Because proteins can be thermally unstable, and if you make them less stable, those proteins unravel. So you look for mutants which are normal at one temperature and which at high temperature look like this because they're defective in a gene necessary for reproduction. That's the process I went through, and you can identify genes that are necessary for the cell cycle. In my original work, I identified 30 such genes, cell division cycle genes. I did some crude statistics on that, which I won't try to explain to you, and predicted that between 250 and 400 such genes would exist. I now know, because we've now sequenced the fission yeast genome, we've deleted every single gene in that genome, I now know there's about 300 genes. So at this early stage, we knew we had 10% of the genes, but there was still 90% to find. Now I want to just show you what a fission yeast looks like, and you can see it there on the left. And before you get too excited by it, I should say that it is a particularly useless yeast. In the town of Carlsberg, the budding yeast is sensibly worked on, and that makes decent beer, it makes bread, it makes decent wine. This yeast only makes a rather disgusting beer that is drunk in Africa, and a very cheap spirit that is drunk in the Caribbean. And having made beer with it, I don't recommend it to anybody. But I put here on the right the earliest picture I could find of fission yeast, which is in a book by the Carlsberg brewer Jacobsen, published in 1896. And since it was drawn in Copenhagen, I thought you might like to see it. So this is the earliest picture that I have of fission yeast from 1896. So this is the organism that I subject to this approach. I had to be taught how to do this, and I was taught by two of my mentors and teachers, and I want to mention both of them. One of them was Murdoch Mitchison, who was from Scotland. You can tell that there. This was a Burns night. This is my wife, actually, still my wife, doing the washing up, as is my professor. And she always remarks when she sees this photograph, there's washing up to be done and there's no sign of Paul. I think it's most unfair, but I feel I have to say it. Then I spent, here he was interested in cell physiology, but was not a geneticist, but I learned genetics from the founder of fission yeast genetics. He was the founder of fission yeast physiology, Urs Leopold, who, as you can see here, was Swiss, and this was actually his retirement party, and he's got an alpine horn. What I want to tell you is that I worked in these two professors' labs in total for 7 years. I published about 15 papers, and they did not put their name on any paper because they did not think they contributed enough to the paper. They were happy simply to provide the support to a young investigator. Huge, huge generosity, which I just want to point out. They're both dead now, and acknowledge the great support that they gave me when I was that age. Now, I explained to you the search for cell cycle mutants. I just want to schematize that here. This is a graph of time, and this is cells increasing in size. And I want you to focus here. This is a cell that's just been born, and it grows, and it divides, and this is the normal cell cycle. It undergoes this process. If you cannot complete a process important for that cell reproductive process, then the cells will continue increasing in mass and give us those elongated cells that I showed you. So that's just to repeat the argument that I've already said. And this is what they looked like, and this is the first paper that was described from 1976. This is a graduate student who was working with me, Kim Naismith, These are real pictures of wild type, that is normal cells, fission yeast, and here you'll see a couple of those highly elongated cells, defective in a CDC gene required, one of those 30, required for cell reproduction. This one here is actually defective in the process of division itself and fills up with disorganized material. So at this point, in the mid-1970s, I felt we had a way of at least identifying genes required for the process that we were interested in, cell reproduction. But actually, I was interested more in what controlled the process of cell reproduction. And this requires me to say a little bit about controls, which is one of these words we use very loosely, and it is good to give some greater thought to what we mean. And I'm going to think about it in two different ways that I'd like to communicate to you. One way is a control is something which is rate limiting for the process which you're interested in. And if I can give you an example, if you took a car and you were interested in what controlled the speed of the car, then of course the accelerator, the throttle, would be the component you would look for. If you altered the throttle, then you could speed up the car, whereas, for example, if you took off the door of the car, it would have no effect. So you would look for mutants that could speed up the car. So what would such a mutant look like if it wasn't a car but a yeast cell? Back to this diagram, but the other part that I didn't speak to you about. If we now have the normal cell cycle and it takes this amount of time to undergo all the events of the cycle, the cell will grow from here to here before it divides. If you now have a mutant in a gene which is defective in a rate-limiting step, in a way that it will speed up a rate-limiting step, then potentially, because you don't know if such a control exists, potentially, instead of taking this amount of time to complete cell division, let's say in the extremis, it can complete all the processes in this shorter time, then what you would get is a cell that would divide at a small size, the complete opposite to here. Now, this is likely to be much rarer, but is one of the things to look for in control. And I'm going to talk about that, and then I'm going to talk about another control in a moment. So if this is true, and once again this is a step of the imagination, then you should look for cells that are dividing at a small size, but in other ways are normal. Now, I'd like to tell you that I went through that logical thinking and then started to look for such cells. But I have to admit to you, I did nothing of the sort. I was actually simply screening, looking for cells looking like this, when, by accident, I saw a clone of cells looking like this. In other words, I never thought of this until nature gave it to me. And when nature gave it to me, I was quite quick to understand that it was important, but I would have never have thought about it until I saw it under the microscope. And I mention that because of the importance of serendipity and chance in scientific investigation, and the importance of being aware of what the world has out there rather than our own mental constructs of the world. Because if you are too theoretically based, you only find what you have previously thought of. If, on the other hand, you are open for what's out there, you can find new things that you haven't thought of. And so you not only need leaps of imagination of what you have thought about, but you need to pay attention to what's out there in nature so you keep grounded. So that's the second thing I wanted to emphasize. So I found such mutants. I got very excited about it. I showed it all to all my colleagues at the time, but I must admit they just thought I was in one of my over-excitable states, which they'd been subject to many times in the past. And it did take a bit of time to persuade them that indeed this was interesting. And here was one of those colleagues, Peter Fantes, who helped me think a lot about this particular problem. He's down at the bottom right. And this is the mutant that I first saw. I discovered it working in Edinburgh in Scotland, and so I called it a wee mutant, wee being the Scottish word for small. I did think it was quite funny at the time, which is in 1975. I have to admit that the joke is wearing a bit thin now, 40 years later. But anyway, I still have to call it a wee mutant. By chance, the very first mutant which I found by chance was temperature sensitive. I wasn't looking for temperature sensitive, it just happened to be temperature sensitive at low temperature like this, at high temperature like this. And this allowed me to do a very useful experiment, which is simply to shift from low temperature to high temperature. And therefore, that allowed me to work out where in the cell cycle it acted. Because if it acted early in the cycle, it would take some time before cells started dividing at a small size. If later, then it would take it occur very quickly. This is such an experiment. The shift up was here. The time for the cell cycle was 2 to 3 hours. And this is the percentage of dividing cells, of mitotic cells. And you'll see that occurs within about 40 minutes to 1 hour of shift up. So that allowed me to conclude that this gene, which I called WEE1, was acting towards the end of the cell cycle just before mitosis. So this incredibly simple experiment told us the following things. There was indeed a rate-limiting step for regulating the cell cycle. It didn't have to be, but there was. Secondly, we had one gene involved in it, WEE1. Thirdly, that rate-limiting step was at the end of that G2 cap and controlled the onset of mitosis. So it told you a lot with a very, very simple experiment. Now, the next experiment is a bit complicated. So you can have a little sleep, if you like, for the next couple of slides, and I'll wake you up if it's a bit complicated, because it doesn't matter. The main reason I want to show you is that it showed what I looked like at the time. And this is my friend Pierre Thurio, and this is me, okay? Over here on the left, looking a bit, if you're old enough to know, like Frank Zappa at the time. You can see the drug fumes coming off my friend Pierre Thurio's head here in his caftan. I'm, as usual, talking. Now, this was a paper published in 1980. What it was to show was that this WEE1 gene encoded an inhibitor of the process from G2 to mitosis. I don't want to go through the logic of it, but for those of you who are more biological, the experiment showed is that there were mutations in WEE1 which could be suppressed by a nonsense suppressor, that's a tRNA that could act as through a read-through codon, and that suggested that those mutations were truncated proteins, and truncated proteins, we argued, were likely to have a loss of function. This is the sort of ridiculous experiment we had to do in the 1980s. We can now do it in a much, much better way, and it is, of course, flawed anyway in the argument. But as a consequence of it, we concluded that WEE1 was likely to be an inhibitor of the rate-limiting step, and indeed, that turned out to be correct. So we had one gene responsible which acted in the control. Now, I was quite excited by this because I felt we were beginning to get into the system. So I then spent a year or so isolating more of these mutants. Now, they're not easy to get. There's no way to select for them. You just have to visually examine every clone under the microscope. And I decided I would collect 50 mutants to see how many genes they would define. And every time I got one, I would cross it to the original WEE1 mutant to see if it was the same gene or a different gene. And I got to mutant 48, and they were all in the same gene. So this was a bit depressing, you know. I spent a year, and I still only had one gene. The 49th, remember I was going to stop at 50, the 49th, I found on a plate covered with a filamentous fungus. Now, some of you may be familiar with this. A filamentous fungus grows rapidly over the whole of the jelly plate, the agar plate that one grows these cells. And this meant that it was extremely difficult to recover the mutant and get it separate from the fungus. So I was tired. It was a Friday evening. It was raining in Edinburgh. And I threw it into the rubbish bin and said, I can't be bothered with this. I went home on my bicycle. I got wet. I had my tea. And about 7 o'clock in the evening, I started feeling guilty. I kept thinking of this plate in the rubbish bin. and thought, well, maybe it was important. So eventually I cycled back, and it was dark now and raining even more, and it being, the United Kingdom in the 1970s, nobody had emptied the rubbish, so I could go back and retrieve the plate, the jelly plate, which I did from the rubbish bin, and I started to micro-dissect it away. It took about 2 weeks to clean it up because you always took a bit of fungus across. I eventually got it, and you know what I'm going to say. This is the only mutant out of the 50 that I isolated which was not a mutant of WEE1. It was a separate gene. So that made me realize there was a second gene that was actually important, a gene which I called WEE2. Now, WEE2, and again, you don't have to, I'll leave it up here, but I'm not going to take you through this. This is, some of you who understand this stuff can look at it, but the rest of you don't have to look at it. WEE2 turned out to be dominant. That is to say, when you made a diploid and combined it with a wild-type allele of WEE2, it actually was still advanced into mitosis at a small size. And so geneticists usually conclude when you have dominance that you now have a positive acting gene. WEE1 is negative, and WEE2 looked as if it was positive. That is to say that WEE2 encoded a gene product that was needed for the onset of mitosis, but if you made a hyperactive version of it, it would go into mitosis early. Now, I started to think about that and then thought, well, if it's needed for mitosis, then if you knock that gene out, you will not go into mitosis at all, and you'll get those highly elongated cells. So although I only had 30 genes, if I was lucky, and if you're a scientist, you always have to think you're lucky, even when you're not, that if I was to cross that WEE2 mutation to all my 30 CDC gene mutations, maybe one of them was the same gene. So I did those 30 crosses, and it turned out that one of them, a gene called CDC2, was very tightly linked to WEE2, so tightly linked that it looked like the same gene. Now, although it looked like it, I must say, and again this is a bit more technical, I didn't think that was good enough, so I then proceeded to isolate 10 or 12, I should say, new CDC2 mutants just by visual screening. I made a fine structure map, which is over here, of the CDC2 gene, again for those familiar with it, of the sort that Seymour Benzer did with phage, and mapped the WEE mutant allele in the middle of it. So that told us that WEE2 was the same gene as CDC2. So now we had two components acting in the rate-limiting step controlling onset of mitosis. A negative one coded by WEE1, a positive one coded by CDC2. I was enormously excited by this. The problem was the rest of the world wasn't enormously excited by it, and I was looking for a job. And I kept being interviewed for jobs and failing to get them. And the main reason was that a friend of mine, quite a few years older, Lee Hartwell, who got the Nobel Prize at the same time as myself, was also working on the cell cycle in a different yeast, and he had identified a different type of control which acted at the other end of the cycle, at the beginning of the cycle. And he had identified a control he called start, which is the point at which the cell makes the decision to undergo the process of reproduction. And this acted early in the cycle, before DNA replication, during G1. And the way he established that was to use the CDC mutants, which he had isolated, block cells at different stages around this cycle, and then ask, could these cells do something different if asked to do so, instead of reproducing themselves? And what they could do is to conjugate with another yeast cell. And it turned out that if you were blocked early in the cycle, you could conjugate, and if you were blocked later in the cycle, you couldn't. So he called that start, the point where you commit to the mitotic cycle. I'm sure you get the general sense. So I thought this was extremely popular at the time. So I thought, well, if I want to get a job, I could do the same experiment, and it's a bit of a me too experiment, but people would be interested. Maybe I could get a job, and that would be good. So I went and did the experiment, very straightforward, blocked the cells, got some nice results, showed that there was a gene, CDC10, that blocked before start, and I had about 100% conjugation when they were blocked there, so they were uncommitted. And then I did a whole variety of other blocks later, and the conjugation was low, between always lower than 5%, 0.5%, 2 to 3%. And then I decided I would do the control experiment. And the control experiment was my best studied gene, which was the CDC2 gene, which I knew controlled the onset of mitosis. I did the experiment, and I was looking, of course, for a result of less than 5%. That is, it didn't commit. And I got an answer. Well, I used two experiments here, 31% and 19%. Now, I know I'm a biologist and therefore not very quantitative, but even I realized that was difficult to make 5%. So, usual thing as biologists do when faced with a problem like this, I did it again. I ended up with 20%. Next thing biologists do faced with a problem like this is buy bigger thermometers to measure the temperature of the water bath in which you're doing the experiment to make sure you get it exactly right. I bought the better thermometers. I did the experiment again, 20%. Then you put it away in a drawer for a while. For a week or two, you do it again, hoping, that the moon is in the right part of the sky. Do the experiment again, and you get 20%. Then you go through a period of depression, and I had a dream. Now, in my dream, which I woke up in a cold sweat from, I had on one shoulder an angel, and on the other shoulder a devil. And the devil was whispering in my ear, and the devil was saying, look, you've done all these experiments, and it's a nice paper, and everybody will be interested in it, and you'll maybe get a job if you do that. And there's only one result which doesn't fit with that, and that's the result with CDC2. But actually, there's only two people in the world who know that you've done that experiment. Your technician, Yvonne Bissett up there, and you. So you could sort of forget it. And then you could publish your paper. So that was one way. Meanwhile, the angel was whispering in my other ear and saying, you can't do that, Paul, he said. I know that if you submit this paper with that result, it will be rejected and you won't be able to get it published. And I know that means you won't get a job. And I know that means you won't be able to pay your mortgage and your 2 babies will die on the streets. But science is the pursuit of truth, and you cannot do that. So I woke up in my cold sweat and thought there has to be a third way. And the third way was actually to believe the result. This is something else I wanted to bring to your attention. When you go into an experiment with a preconceived idea, which was you either get a result over 80 or 90% or you get a result under 5%, when you get something that doesn't fit, you assume the experiment is wrong. And after a while, you have to start thinking, what if it's right? And over the previous 4 or 5 months, I'd simply assumed I'd done it wrong. So I then went away and I thought for several days, because my mind's a bit slow, as to what might be the explanation, and I came up with the crazy idea. And the crazy idea was, although I thought CDC2 only acted at the G2 to M transition, where it was acting in a rate-limiting step, maybe it also acted at start, and because G1 was very short, when I shifted to the high temperature to arrest cells at different stages in the cycle, most of them would block here, but about 20% could block here, and they would conjugate. Quite a simple hypothesis, and it turned out to be true. When I synchronized cells, made them all go into this part of the cycle, and then shifted up, 100% of them conjugated, because they blocked at the right point. And so that meant CDC2 acted at the 2 major control points in the cell cycle, both of them. And those events are very different. It was rate limiting for overall cell cycle progression and controlled the onset of mitosis. And it controlled the commitment to the cell cycle, which also controls the onset of S phase. And you may remember from my introduction, I said that these two events are the major events of the cell cycle. Now, when I got that result, I thought, now I just have to commit the lab to finding out what it is that CDC2 does, and I will understand how the cell cycle works in yeast. Now, how do you do that? Because at this moment, everything I've told you is rather abstract. You know, we've just talked about genetics and genes and identifying genes. Well, it was about this time, the early 1980s, that molecular genetics was coming along. So I could now start doing molecular experiments. But to do that, one had to introduce DNA molecules into yeast, and you had to be able to select for cells that had taken up that DNA, and you had to get what we call DNA plasmids to grow inside of the yeast cells. And this was about the time I came to work in Copenhagen, and I'd got the experiments to work in the University of Sussex. I came here, and I think every experiment failed for about 3 months. I had to do them in the Carlsberg brewery because of the containment facilities. And I didn't realize that the solutions we'd made up in Sussex, we'd made up incorrectly, and it was the incorrect solutions that worked. And when I duplicated them here, they didn't work. And it was only when I exchanged all the solutions I found that out, and found to get it to work, I actually had to use a different solution from the one that I thought should work. This is crazy stuff, but it's just how science works. So this was transformation, which I worked out with a postdoc in my lab called David Beach. So now what we could do with that process in place is clone the CDC2 gene. And this is a fission yeast cell. It's defective in CDC2. This is a gene library of fission yeast, and it contains one of them, it's 5,000 genes, remember, contains the CDC2 gene. We transform it in, and the yeast cell that takes up a plasmid with that gene takes up a plasmid with that gene, makes the CDC2 protein, rescues this defect, and the cells can divide and form colonies on the plate, whereas the original one cannot. You're using complementation, as we call it, to clone the gene. So that experiment was done. The gene was isolated. It was sequenced. It took 1 year and 3 months to sequence 2,000 nucleotides, for those who are familiar with this. We could now do it in about 1.5 seconds. And subsequent analysis, which I won't go into, and was long and laborious, indicated that this gene encoded an enzyme which was a protein kinase. What is a protein kinase? A protein kinase is an enzyme that puts a phosphate, which is a bulky residue, negatively charged, onto another protein. And when that bulky phosphate residue goes onto another protein, it changes the properties of that protein. So this is a very good switch molecule, because a protein kinase like that can phosphorylate many different proteins, and it can activate many different processes. So it's a very, very good switch. I'm just trying to show it here. You put this red blob on this molecule here, it changes the configuration so the green molecule can get in there. This is just to show you how this, in principle, can work. So with that experiment, and we showed that CDC2 had protein kinase activity, with that experiment, we could now suggest that CDC2, major rate-limiting step for the cell cycle, encoded a protein kinase that phosphorylated other proteins. So we measured protein kinase activity as it goes through the cell cycle, and this is an experiment done by Sergio Moreno in my lab, now worked in Salamanca. This is the next phase of my life over here. Now looking like, Mrs. Doubtfire or something. Anyway, and this was the protein kinase activity, which was lowish through much of the cycle, and then comes to a peak just as the cells underwent mitosis. So we could now postulate, we had a molecular mechanism, that the CDC2 protein kinase phosphorylating other proteins does it by the kinase being activated at the onset of mitosis. And this is the major rate-limiting step for cell cycle progression. Another postdoc in my lab, Paul Russell, because all these efforts are now entirely collaborative, and these all brought skill sets that I didn't have into the laboratory. And in several papers here, Paul Russell, who now is working on the west coast of America in California, identified 2 regulators of CDC2, WEE1, you remember WEE1, which is another protein kinase that phosphorylates CDC2, CDC25, which takes that phosphate off. So what we now postulated was that the way in which the CDC2 protein kinase is activated is in fact by WEE1 inhibiting CDC2 with phosphorylation and CDC25 taking that phosphate off. And that was confirmed by Kathy Gould, who came to my lab, now also working in America, and she identified an amino acid residue in the CDC2 protein, phosphatyrosine, that becomes phosphorylated in this control. So the whole thing was a very simple switch. And the final piece of molecular stuff was that another graduate student in my lab, this time Ian Hagen, showed that a protein that touched CDC2, one that we called CDC13, looked like a molecule that had been found in sea urchin eggs called cyclin. And this was a protein that had been linked with the cell cycle. So this is where we were with fission yeast. What we had was CDC2 controlling onset of mitosis and working in a way we didn't understand, the S phase, complexed with another protein, CDC13, regulated by a switch controlled by the WEE1 protein kinase and the CDC25 phosphatase. Now, the problem with all of this was it was yeast. And frankly, who is interested in yeast? Now, as it happens, I'm interested in yeast, but not many other people were interested in yeast. And I found myself working in a cancer research institute, and most of my colleagues, the only thing they were interested in was keeping me out of their laboratory in case my yeast contaminated their precious human tissue cultures. And I felt a little bit like a sort of leper in the laboratory. So I thought I have to do something which shows these people that I'm relevant. And I thought the only way I can show I'm relevant is actually to see whether CDC2 exists in humans. And a postdoc, I'm going to show you a picture of her in a moment, came to my lab and said she wanted a difficult project. So I said, okay, try and isolate the human CDC2 gene. Now, this may not sound so difficult today, but as you've heard, yeast and humans diverged at least 1 billion years ago. And just to put that in case you're not good on geology, dinosaurs went extinct a mere eye blink ago, 65 million years ago, and animals only appeared about 600 million years ago. We are deep in geological time. So there was no reason to think that a CDC2 gene existed in humans. It was another leap of the imagination. So she started looking for it. And by the way, there's no human genome sequence or anything like that. That's 20, it's 15 years later, so you can forget that approach, which is what we do today. So we tried conventional techniques. Conventional techniques are to look for a human gene with structural similarity to that fission yeast CDC2 gene. Structural similarity means sequence similarity, which you could detect by hybridization, or maybe that it makes a protein which can be detected by the same antibody, showing that the proteins are related. So we did those experiments over about a year and a half, and we isolated human protein kinases, which had sequence similarity to CDC2. But the problem was, by that time, everybody thought there were 500 to 1,000 protein kinases in human cells, so how on earth did we know whether we had the right protein kinase? So, on the edge of giving this project up, we decided to use this approach that I've already explained to you, with one difference. This is cloning by complementation. So here we have the fission yeast CDC2 TS mutant again, and we are going to transform it this time with a human cDNA library. That is to say, a human library of genes to see whether there is a gene in humans that can rescue this. What this looks for is not structural similarity, but functional equivalence. That is, a gene that does the same job. Now, at this time, there was only one such library present in the world. It had been made by Paul Berg in California, and he'd only made it a few months before. And once again, generosity speaks. I wrote to him, I asked for the library. He said, have it, do whatever experiment you like. Just very, very generous, yet again. We did the experiment. It had no right to work, but it did. We put this into the cells. We found a gene that worked. The gene was isolated, and it turned out to be the human equivalent of CDC2. And this was done by Melanie Lee in my lab, postdoc here, very important experiment. She went on to go into the biotech industry, was very successful. In 1 year, she earned more than I've earned all my life, actually. I just obviously did the wrong career, but there we are. And she was very, very successful, and I am always grateful for her because she did this crazy experiment and she did a crazy project and then she did a crazy experiment and it all came together. Just really good. And this just to show you the sequence identity, 1987, between human and fission yeast, and I remember it today. Fission yeast, the protein is 297 amino acids long, human is 298, just one amino acid difference, and there was 61% identity at the amino acid level, and this is 1 billion years of divergence. Highly, highly converged, and the conclusion we came to was that, in fact, the human CDC2 gene exists, and that, we argued, means that the human cell cycle is controlled in the same way as the yeast cell cycle, and if humans and yeasts are controlled that way, it means every living thing in between is also controlled in the same way. This is, by the way, a picture I took of the first yeast clones growing with that human CDC2 gene in it, and the ones that are very elongated are cells where the plasmid has been lost, and so the cells can no longer actually divide. We went on and isolated, you don't have to read this, the equivalent CDC2 genes in mice, in chicken, in flies. Every organism we looked at has got this CDC2 gene. Now, everything I've talked about up until now is the control of mitosis. CDC2 also controls the onset of S phase at the beginning of the cycle. I won't say anything about that because I'm going to end my talk with another control which CDC2 does, which was completely unexpected, because I was also interested in a problem described here. And that is that every cell cycle, there is only one round of DNA replication. That's crucial. When a cell is born, it undergoes DNA replication, and then it undergoes mitosis. If it undergoes more than one round of replication, then you'll have twice as many chromosomes as you should have at mitosis, and the ploidy of the cell will change, or you'll get a defective mitosis. So there is a very strong cell cycle control which ensures that there is only one round of DNA replication, the Watson and Crick thing, every cell cycle. And so this is another major control which I was interested in the cell cycle. To get at this, I went for another mutant screen which looked for cells which would actually undergo a second round of replication or more instead of going into mitosis. And used various approaches, but one approach was to look for mutants which actually would induce this. And did that, and I'm shortening this story, and found a mutant that looked like this. This is cells which are stained for DNA, and this is a wild-type cell, and this is the mutant. And you can see that here the cells are very small, here they're very big, but look at that nucleus. This has got a huge amount of DNA in it because this mutant, just when you shift it to the high temperature, just undergoes round and rounds of DNA replication, one after another, when it's switched off. And here I've done a measurement of the DNA content, and this particular cell is actually, we've deleted the gene, and it goes up to 64 copies of DNA. So it's repeated rounds of DNA, which means that the cell is locked into repeated rounds of DNA replication. When we investigated what the gene was, it was CDC13, the cyclin component of CDC2. And this was carried out by Jackie Hales, a long-standing collaborator in my lab, who actually just retired this last year. So what this meant is that this CDC2, cyclin-dependent kinase, as they're now called, or CDK, had a third role that was completely unknown, which is that during G2, it prevents reinitiation of S phase. And when you delete the G2M form of CDK, the cells don't go into mitosis, they lose that inhibition, and the cells get promoted to go back into G1 and to re-replicate and re-replicate and re-replicate. And why that's important is now the CDK is controlling onset of S phase and the decision. It's rate limiting for the onset of mitosis, the second major point, and it's in the third major control of the cell cycle, which ensures there's only one S phase in each cell cycle. Because as soon as the CDK appears, preparing for mitosis, you cannot undergo another round of S phase. A beautifully simple system which explains all the main properties of the cell cycle and which involves only one major component, the cyclin-dependent kinase encoded by CDC2. And that's really drawn out here. And this is actually somewhat more recent work, but what we propose is a very simple model. When a cell is born, it's born with no CDK activity. The amount of CDK activity is represented by the redness. You get a small amount of CDK activity and you enter into S phase. You get more CDK activity, you inhibit S phase. You get more CDK activity, you go into mitosis, and you get out of mitosis by destroying all the CDK activity, and you start again. You could not imagine a simpler model. There's different ways of regulating it, and a major regulation here is through the Wee1 negative phosphorylation. I've rolled the cycle out here so we can see the CDK activity going from a low level to a high level, and we showed that you only need a low level to undergo S phase, you need a high level to undergo mitosis, and when you're in G2, you're inhibiting S phase. If you let the CDK activity rise, you go into mitosis. If you now destroy the activity and knock it down here and then let it rise, you go into S phase. And so that shows that it has that third role during G2. It also tells you, incidentally, that there is no arrow of time in the cell cycle. The direction that you travel is determined entirely by this CDK activity. It's not hardwired of S phase mitosis cell division. It's actually, you can switch events around simply by manipulating the CDK activity. So let's summarize where we are. CDK is actually the major control of all eukaryotic cell cycles that have been examined. Increasing activity drives the onset of S phase and commits you to the cell cycle. More activity inhibits S phase, and that ensures, as you're preparing for mitosis, that there's only one S phase per cell cycle, the important control I just mentioned. When you get more activity, you initiate mitosis, and then you exit mitosis and complete cell division by destroying the activity and starting again. And you can do all of that with a single cyclin-dependent kinase. And the second point is, basically, the control is the same in all eukaryotes. Now we're less surprised by such conservation, as was mentioned in the introduction. At this time, it really was a bit of surprise. However, perhaps we shouldn't have been so surprised, because if you dig back to the 19th century, I found a quotation from Theodor Schwann, who was one of the great proponents, Schleiden and Schwann, of the cell theory. He wrote in German, but this is my last slide, and it's translated into English. What he said was this: we have seen that all organisms are composed of essentially like parts, namely of cells. That's a statement of the cell theory. Then, listen to this, that these cells are formed and grow in accordance with essentially the same laws. Hence, that these processes must everywhere result from the operation of the same forces. It's exactly what, of course, it took another 150 years to actually establish. So we shouldn't have been quite so surprised. We were anticipated by one of the first proper cell biologists in the beginning of the 19th century. Thank you very much. Thank you.
Yeah.
Good for you. One at the back. Off you go.
Yeah. You mentioned this time symmetry, that it could have gone backward,
Yes.
the wrong way around. Does it mean that the time symmetry is just broken because when the CDK disappears, it disappears very fast and nothing happens?
It means when the CDK disappears, that sets the beginning of the clock, essentially. Yeah. The gradual rise does take some time, but as you've indicated, the disappearance is very rapid. Probably has to be rapid to set yourself from undergoing the events to starting again. We incidentally have some idea of what the molecular basis of what's happening there is, but really the principle is just very, very simple. Very simple. Just a gradual rise of CDK activity.
Yes, but it's very important that it does it rapidly. Otherwise, it would do everything backward.
Correct. So they kill it very rapidly, and without that, it could give some problems. Now, I have tried playing around with doing it slower, which you can do with inhibitors, but the results have not been very clean. So, and I suspect, actually, it's just messing up in the way that you're predicting.
Just a philosophical question.
Hello.
When you explain about the cell as a part of the total representation of life and cells, what about, could you see any connection between fractals where you have a small representation of a bigger connection?
You mean fractal...
mathematical fractals and...
I'm actually quite interested in chaos and fractals, and I think it is illuminating for certain biological phenomena, but I don't think it's illuminating for what I've spoken about yet, at least for today. I think that the similarity in a whole range of organisms at a completely different scale simply reflects that at the level of the cell, it's a rather simple and highly conserved system. So I don't think fractals are useful for that, although I do think thinking about chaos is useful in certain other biological phenomena.
So this is a beautiful, simple life cycle system, but one question brings up a new question. So what makes the CDK activity to disappear and start a new cycle?
Yeah. So I haven't worked so much on that, but it's pretty well understood. You'll recall I said there were two components to the cyclin-dependent kinase. One of them is the protein kinase itself, which I mainly talked about. CDC2 is the archetype. The other component I only briefly mentioned and was worked on by my colleague Tim Hunt is a cyclin, and that's why they're called cyclin-dependent kinases. The cyclins are destroyed by very targeted proteolysis that chews up the cyclin protein and completely destroys the activity. So that's the way in which you destroy the activity, and it occurs, I mean, as the gentleman in front was asking, extremely rapidly.
In line with the same, and probably you gave part of it, CDC2 regulation of cell cycle is obvious, but have you a guess of how many further kinds of control there exist? Because I think the total cell machinery cannot rely on only one control, but to fine adjust and so, have you any guess? You had 300 genes in your initial talk.
Of course. What I think is that the CDK system is in fact the major core regulator. But there's several things to say to modify that statement. The first is, I've talked about what we call eukaryotic cells. I've not talked about prokaryotic cells such as bacteria, because they do not have this control system. So they undergo a cell cycle with the same biological issues to deal with, the same biological problems, but they solve it in different ways. So we're not talking at all about bacteria, we're only talking about, well, of course, all animals, plants and fungi and so on, which are controlled by this system. Second point to be said is that, of course, there are controls that act upstream of this, that in more complicated organisms particularly, feed in all sorts of inputs. Because in our human body, we have to control, for example, the cell cycle in very precise ways during development, in tissue repair, in a whole set of other developmental circumstances, and the CDK system has to be regulated upstream of that. Yeast, it's simplified, and so you don't have so many controls. A third thing to say is that although this may be the major core regulator, within the events themselves, within DNA replication, within mitosis itself, there will be lots of smaller controls to ensure orderly progression through these events. And these are the sorts of things that will be revealed by study of the 300 genes that I mentioned and that you referred to. But the major key regulator does seem to be this CDK cycle that I've described.
In some cases, in some cases
Hello, where are you?
There.
Oh, yeah.
In some cases, a cell may stop dividing and simply continue growing. Has there been any work done on the mechanism behind this? On why the CDK is not broken down?
I think I got the question. Are you asking, has any work been done on the growth of the cells and what controls the growth so that they keep growing?
Well, yeah, but has any work been done on why the CDK may in some cases not be broken down so that the cell will continue growing and not divide again? Right.
So this is because the controls over growth, that is the ability to make RNA protein cell mass, are quite different from the controls that ensure the reproduction of the cell, that is a division from 1 to 2. And actually, rather small amount is known about what controls overall growth. We know something about it, and I've been trying to investigate it in the last 5 years without actually great progress. But I think it's a very important control, and is actually crucial for diseases like cancer, because if you don't grow, even though you're reproducing, then that will not give you cancer, because the cells will just get smaller and smaller, and they won't be a problem. So growth control is actually absolutely critical for understanding cancer. We know many of the genes that influence it, but we don't have a clear picture, like we do with the cell cycle, of how that is regulated in an overall way. There's good leads, but it's not yet fully established. But it's an important problem, which I would have liked to have got some inroads in with yeast, with fission yeast, but have not managed to do so.
You told us, of course, that you have an enzyme putting phosphates on other proteins, but you didn't talk very much about those targets. Could you very shortly tell us how many are known now that are targets and what kind of?
Yes, I can, because I have a graduate student who is identifying the targets, and he's using a mass spectrophotometer to identify proteins that become phosphorylated and phosphorylated at the right time. We have, in those studies, already identified about 120 substrates phosphorylated at mitosis, and only about 15 that are phosphorylated at S phase, so a very big difference. And we're now analyzing them to see which the proteins are and what may be important. But curiously, although it's been worked on somewhat, there's very little complete understanding of those targets, as you've hinted. But I'm really, really optimistic that we will, in the next year or two, get a very good catalogue of that. We've got it working, he has got it working really well.
Do you know whether CDK has similar effects on meiosis as well as mitosis?
Very good question. So Meiosis, which I'll just remind the audience, is a modified cell cycle that occurs when gametes are made. And what's different about it compared with a mitotic cycle is that after S phase, you have 2 M phases. So you can reduce a diploid DNA content to a haploid. That's half the amount of DNA because you undergo 2 divisions instead of 1 division, one M phase division. So we have, curiously, that you should ask this question, I have a postdoc looking at all the cyclins in the fission yeast cell and which ones are needed for meiosis. And studies showed that it was thought that 6 different cyclins were probably required to give you a range of different CDKs. But by genetic manipulation and simplifying the system, we've basically reduced it to a single cyclin-dependent kinase and got very efficient meiosis, and it's working in a very much similar way to mitosis. We don't understand it quite so well, but despite the apparent complexity, it looks as if it's going to be quite a simple solution. So work in progress, but I think we've got basically the same answer as the mitotic cycle.
In certain societies, especially in America, there is this debate with intelligent design and nature. You got a Nobel Prize. I guess you've analyzed this. What's your position when you find such a beauty in nature in comparison to the question before with fractile? What's your position? How could this process come in place? And it seems as though there is a higher order for this process to continue.
Well, there's no doubt that there's a higher order in the control of the system, but there's no evidence, of course, of intelligent design in that, none whatsoever. And there's things we don't understand about evolutionary change, of course. I would say that this system is not so difficult to imagine how it came together. I'll say a few words about it. It's obviously more complicated when we're dealing with complex organisms such as our cells, because the modern cell cycle actually has, and I didn't emphasize this because I didn't have time, although I hinted at it just now with a meiotic question, the modern cell cycle, as I shall call it, has a range of different cyclin-dependent kinases that catalyze different steps in the process. Now, what we have found is by simplifying the system that we can drive the whole thing with just one cyclin-dependent kinase. We've had to genetically engineer that, but we've managed to do it. And that makes me think that the very early eukaryotes, maybe 1.5 billion years ago, had only a single cyclin-dependent kinase that acted more or less as we did here. That may not have been completely efficient. And so then you add on to it other cyclin-dependent kinases to make it a little bit better, evolving the complexity that you're talking about. So at the end, you actually have quite a complex process. But the evidence that you can simplify it again is strongly suggestive, in my view, that in fact this incremental increase in complexity is the best solution we have, at least for systems like this.
Hi, so my question is, all of the experiments you've done and the signals you've observed were based on averaging the signal over a cell population, a cell culture, right? So I'm guessing that the various effects you're observing must have a distribution across the given cell population. So my question is, are all mutations that you observe display the same sort of distribution in the sense, like, are they as heterogeneous within the cell population? And would that hint at mechanism by which life is constraining the randomness of the chemical reaction on which it's basing it on?
Yeah, so this is a good question. Actually, not all the experiments were done just on populations, though you're quite right that many were. Some of them were single-cell experiments which reveal the variability that you're talking about. Now, one thing I haven't sort of emphasized but comes out of the study is that cells progress through the cell cycle when they reach a certain cell mass. So they need a cell mass to undergo S phase, another cell mass to undergo mitosis. And by single-cell studies, we can look at the variability of that to get some sense of the issue that you've talked about. And I've just started a graduate student on a project where we can analyze many thousands of single cells in a new device called an image stream, a bit like a fax if you're familiar with these things, as I suspect you are. And we are trying to work out the variability that occurs at different cell cycle transitions, because this is, of course, something that's becoming very popular, and in particular to see whether the control proteins have a very limited variability when they undergo the transition. So our intention is to assay all the control proteins we've identified and measure their levels in a population of cells, but individual cell by cell, to see whether they are actually restrained at the major transitions. So we're trying to get into that problem initially by that approach.
Yes, thank you for a wonderful lecture. I hope it's okay that I ask you a question about something other than your science, but about the science in society agenda.
Yeah.
I wondered if you're talking to other scientists about science communication or the need to talk outside science, about science, what would be your arguments for doing these things? I.e., I guess I'm particularly interested in younger scientists. If you're talking to them about whether they should or should not engage in engagement with the public, what would your arguments be for them to do so?
Well, I'm a huge enthusiast about engagement for the public. I sometimes use a metaphor, which is this, that we scientists have to earn our license to operate, and we should not take that for granted. And what I mean by that is that we are privileged to do what we do, and we can only expect to do it if we have support of society. And to get the support of society, we have to engage with the public. And scientists can sometimes be rather introverted and think any distraction from doing your experiments is to be totally resisted. And my view is, and I'm a complete enthusiast, as you probably gathered, for what I do, is that I spend a significant part of my time trying to engage the public for this reason, and I encourage my colleagues who are any good at it to do the same thing. And I'm particularly concerned about younger scientists who have a flair for doing it, not being encouraged by their supervisors to spend some time to do it, because the supervisor often is driven by wanting to publish something, and they don't see the bigger picture. And I think we really have to defend those individuals who are quite good at communicating and good at engagement, because it's a rare gift. And if somebody has it, then they should be encouraged to spend at least part of their time. This is a very, very serious issue, because if we don't do it, then we will lose our ground to the pseudoscientists or the anti-scientists. We will lose the potential that science has to benefit humankind. So we really do have to work hard at this. And it doesn't even take that much time. I mean, I'm an active scientist. I do it a bit. I'm not talking about becoming, a TV presenter or something of that sort, but simply, if people are good at doing it, let them do it. And it's something we have to train not our junior colleagues, but our senior colleagues, not only to tolerate, but actually to encourage. So I think it's crucially important.